Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 189 for the podcast. Today, I welcome Koenraad Wiedhaup, co-founder and CEO of Leyden Labs, and Clarissa Koch, the company's chief scientific officer. Leyden Labs is pioneering a revolutionary non-vaccine approach to combat respiratory viruses like influenza and coronaviruses. Recently, they published groundbreaking data in Science Translational Medicine, demonstrating that their intranasal antibody spray is safe, well tolerated, and delivers sustained protection right at the virus's entry point, the nose. This innovation addresses the shortcomings of traditional flu vaccines, which average just 13% effectiveness against infection and provide even less for vulnerable groups like the elderly and the immunocompromised. We dive into the science, the company's journey since its 2020 founding, and their recent €50 million European funding boost amid US biotech challenges. I hope you enjoy my conversation with Koenraad and Clarissa of Leyden Labs. Koenraad, welcome. Tell me a little bit about your background in life sciences, Delft and Leiden universities, Columbia Business School, and now co-founding Leyden Labs.
Yes, and great to be here, Dylan. Thank you for hosting us. So I have a background from academia in biotechnology. I'm here in Leiden, sitting quite close to the former alma mater, where I studied life sciences across the universities of Delft and Leiden. I was always intrigued in the application of these different technologies to how can we actually help people to become better, and also then the business side of that. After that, I worked briefly at a biotech company in the Bay Area, which I truly enjoyed in the business development. So already making that jump towards the business side. And then I started at McKinsey, working there in the biotech and life sciences practice, so serving large pharmaceutical companies, but also a lot of smaller, mid-sized biotechs over the time. Was there for about 12 years, both in the US and then in Europe. And on the side, I was always also working with smaller biotechs to help them start up, grow, and was fascinated by that also from my experience at the time that I had in San Francisco at the biotech. And through that, I met my co-founders at Leyden Labs, who were the people behind Crucell. So then I embarked on the next journey towards Leyden Labs.
Clarissa, it's great to have you here as well. You've built your career in immunology and virology. What drew you to join Leyden Labs, and what's your role there?
Yeah, thanks, Dylan. And also seconding Koenraad, thank you so much for having us. So I think if I think back, so really this opportunity came about really in the height of the COVID-19 pandemic. At the time, I was actually working in pulmonary critical care at Northwestern in Chicago at the time. So I was very aware, kind of, if you will, of what we were dealing with and things that we were still kind of facing when it comes to fighting respiratory viral infection. So really, when I heard about Leyden Labs and their approach and really what they were aiming to really achieve, that really spoke to me in kind of in every sense of the way in the context that I was working on respiratory viral infection at the time. So that really, drew me in. And I think to this day, we can still say, we still have very much this need in fighting respiratory viral infection to have more tools in the toolbox, if you will. And that's really what we're trying to achieve here.
Koenraad, Leyden Labs was founded 2020. Tell me a little bit about the origin story there. What's the mission of the company? What are you setting out to do?
Yes, so as I mentioned, I was at that moment at McKinsey, but then COVID hit, all right? And everybody still remembers that vividly. And also, with that, we saw that we don't have enough protection against viruses. In this case, like a new virus, but also in general, where there are millions of people ending up just by simple, quote unquote, flu in the hospital and worse. So that highlighted that need for that additional protection. Now, my co-founders at Leyden Labs were working in that space already for decades, from Crucell, which was later bought by J&J. And they were working on ways to protect much broader against viruses than what we currently do with vaccines. In the first weeks of the pandemic, we came together with the idea of can we give better protection against these viruses broadly, but also then where these viruses come in. So in the upper respiratory tract, the nose, the throat areas, to protect there. And then our founding scientist, Jaap Goudsmit, found an antibody over those decades of research that could protect against all these different viruses for flu. We were able to in-license that from J&J and develop it then intranasally. So then, I was very happy at the work I was doing, but I thought this is really the mission I want to embark on and jointly start Leyden Labs. And then we quickly got fantastic additional people in, Clarissa, of course, being one key there, who had also that mucosal experience where we could complement then that antibody and virology experience as well.
Tell me a little bit about the platform you've got there, the mucosal protection platform. What makes it special? What differentiates it from other things on the market?
Yeah, so what we are in the end developing are nasal sprays that can protect people against different viruses. So, for example, in the flu season, that people can take a daily spray and then be protected against all these different viral strains. Now, currently, how we are protecting people against these viruses is mostly through vaccines, which are very important, but not enough. So flu vaccines only work 30 to 40%, and actually less with people that are highly vulnerable, people that have a lower working immune system. And they have a few key issues. So one is that it protects mostly systemically. We get it as a shot. It's mostly in our system, in our body, and protects there when the virus actually already has replicated in the nasal cavities. Two, they're narrow. So they only protect, these vaccines protect only against a few subtypes of, for example, influenza. That's typically 3 different subtypes. So they don't protect against the full range of such families of viruses. And thirdly, people who need them the most actually benefit the least, because you need your own immune system to have an effective protection with vaccines. And the issue there, of course, people that are mostly vulnerable for the viruses have then the least protection. So what we are trying to do with these nasal sprays, that we want to overcome those issues. So one, that we protect directly in the nasal cavities where these viruses come in, so that we stop the initial infection. And Clarissa can tell much more about that also afterwards. Two, that we protect against full families of viruses, so that it doesn't matter which virus hits us that season, or actually if it's a new outbreak, a new pandemic strain, that we can also protect ourselves there. And three, that it's so-called passive immunity, so we bring antibodies through these nasal sprays directly into the upper respiratory tract, into the nose, into the throat, where vaccines are actually training the body to create these antibodies. We bring them directly in, so that's independent from people's immune status. So with that, trying to get to effective protection where these viruses come in against a broad range of different viruses.
Clarissa, is this a complement to the vaccines that we already have, or is it intended to replace the vaccines altogether?
Great question. I think this is very much, what also Koenraad echoed would be a complement in addition to, I think, as he highlighted, vaccines are really important, not just in this space, but in any space, really. But really, indeed, as he already highlighted, there are some limitations. And I think that's really where we come in. We want to really tap into this unmet niche, if you will. So it's very much, I think, indeed, as he already mentioned, to really add benefit for those that maybe just do not have a full-fledged response to vaccines. But I think also in the case of, for example, as we've seen many times, when we have a mismatch, so where the vaccine is actually not really eliciting a response to the strain that is currently circulating. Now you can think about that. That happens, often on a yearly basis, but also particularly forward-looking. If we're looking at concerns over avian flu or strains that we really haven't really seen yet, that's really where we want to take up, again, very much a complementary niche.
And Koenraad, you mentioned that people who are already immunocompromised or elderly people, they might already have find some really ineffective vaccines out there to protect them. Why is the nasal spray better? Is it just because you're really covering in that area where the viruses enter the body?
Yes, that's one part of it. So that you actually stop these infections already where they start. And with vaccines, that's often kicks in later, where you have already had, a lot of replication of the virus in the nose, in the throat area, and we really stop that there at that gate, as we call it. But then the other very important thing, Dylan, is that for vaccines, you get typically a shot, but you have also some that are intranasal, but it stimulates the own immune system to create antibodies and other responses. But what we are doing is we actually are taking unique antibodies that are very, very broad, as I already mentioned, and we're putting them directly in the nose. So what we're trying to do is, where a vaccine basically trains the soldiers to fight there, we bring the soldiers directly in there, so you don't need the body to do that training. And that's why, where for people that have a lower working immune system, that are immunocompromised or older, or have other diseases, other comorbidities, that this so-called passive immunity, as it is works as effective as for healthy people.
So, Clarissa, maybe you can dive into the science even a little bit more than that. This mucosal immunity, how does delivering the antibody there just make it different to getting an injection or attacking the virus in a different way?
Mm-hmm. Yeah, no, a great question. I think there's a couple things here. I think first and foremost, again, this is really the portal of entry that the virus also, or really, respiratory viruses in general, take to get into our system and really start to replicate. So, for example, for flu, which we've been discussing, that's definitely the point of entry, and therefore really relevant for us to really kind of catch the virus from the get-go, right? So as you can imagine, you get these particles in, and then really already having this nasal spray that's basically coating your upper respiratory tract really kind of is truly the first barrier, if you will, that really prevents the virus from causing any downstream impact down the line. And we've actually already seen that in many of our preclinical data, and we recently published on that as well, that we saw that this concept truly works. And I think, as it is often with science, it seems so straightforward and simple, but it took a lot of years of research to really understand what coding completely a new administration route really could really provide. So seeing that efficacy signal, as we saw, and then also, published on recently, was really truly kind of a really great sort of vote of confidence for us that the approach we're taking here on local administration really is the way to go.
I'm going to come to that published data in just a minute because it is really interesting there. But maybe you can tell me a little bit about the lead candidate that you have, PanFlu. It provides protection against influenza A and B strains. Tell me what it's about and how does it actually help us prepare for the next pandemic, the sort of thing that got you into Leyden Labs in the first place?
Yeah, absolutely. Yeah, so I think very much, the basis of what we're trying to achieve here, and I think Koenraad already alluded to that a little bit, is that we really have the sort of base criteria, if you will, is this breadth of protection. And again, as we already discussed, part of that really is because we see that we always have mismatching. Unfortunately, there's always some sort of mutation or strain that we weren't really expecting in a season. So for us, really, it was critical to see in all of our lead candidates, quite frankly, but particularly indeed also now, since we're discussing the lead candidate for our flu program, to have this breadth from the get-go. And really the way we define that is if we see activity really across every single strain, and that actually also goes beyond A and B. We also demonstrated some data in vitro on C and D. That for us truly kind of was, a definitive answer that this is the breadth we're looking for. Because as you can imagine, and I think a lot of analogies, for me, kind of also, if we look back at the COVID-19 pandemic, we saw kind of similar things happening, if you recall, right? We had to update vaccines every year because there was a new Omicron variant. But we also there very much saw that we therefore needed to be prepared. So you kind of in the moment had to then update. So having something that already has the breadth in any direction possible really means that we don't have to go back and change our lead asset anymore, right? So really having something that can go in any direction, whether it goes, mutates more into a specific H3, for example, if we have an avian flu, H5, we've covered that all. Basically, we've seen that activity holds in whatever direction we look in the influenza strain family.
And this is delivered as a nasal spray every day and throughout what part of the year?
Yeah, so I think for now, and I think this is something also we of course discussed in our publication. We really wanted to prove the efficacy right now. So indeed, we achieved that with daily administration. Again, very much also trying to also gain an understanding of what administration in general in the nose truly entails. As you can imagine, unlike systemic administration, this is a fully exposed portal of entry. So therefore, for us to be able to understand the pharmacokinetics there really took a lot of work. So indeed, for now, we very much took the approach of daily administration. But to be honest, also, we very much were doing this because we wanted to achieve a really sustained trough level, particularly in all the models that we were using to really test our hypothesis, which is a challenge model, whether it's in a mouse model or non-human primates. Really, the basis of these models to prove efficacy of our molecule is to give really high amounts of virus to then be able to demonstrate a statistical significance with our approach. So there, I think this is kind of, if you will, kind of setting the bar the highest possible, but we very much are now looking into, how we can really go down in terms of dosing, but also frequency, because we know in the real world, of course, we're not going to be getting a full, challenge bolus injected directly into the nose.
So let's talk about the data that you recently published. You both had your name on the paper, I think. What were the key findings from that preclinical study?
Yeah, so again, I think maybe I've alluded to some of that, but just to summarize, I think, if we take a step back for us, why this was such an important publication is, everything we've discussed up to now really said that we have a lot of data and understanding within the field on systemic administration of monoclonal antibodies. And the same, of course, goes for vaccination as well. But really, when we started the company, we wanted to understand what does this different administration route, namely going through the nose, what does that really mean, right? And that's really all kind of summarized in the publication. So really where that starts is, of course, understanding the dynamics or the pharmacokinetics of antibody administration. I think a big unknown question that we got a lot, and we had ourselves, was when you administer an antibody, how long does it stick around? And also, does it actually work, right? Because as you can imagine, the nose being kind of the direct portal of entry where we breathe a lot of different things and pathogens in on a daily basis, has kind of a baseline sort of protective system where it also breaks down a lot of things it encounters, right? So we also really had to understand, is an antibody going to work? Is it going to remain intact, right? So that was actually one of the things we were able to really elucidate. So that the antibody was able to really stick around, but also importantly, to still be active, right? We had a lot of characterization studies on our lead candidate, of course, done in the lab and in animal models, but we really wanted to prove that in the human nose, all that activity, all that breadth that I just alluded to earlier still maintained, which we indeed saw. So that was huge. And then I think the second big part for us was to then really tie the presence and being able to achieve a really sustained trough level in the nose to efficacy against those viral infections that we just discussed. So we did that, of course, first and foremost in a mouse model to really kind of validate also that we were maintaining that breadth and seeing that translated into protection against those viral infections. So whether it was an H1N1, an H5N1, or a B strain, we saw the same consistent protection provided through intranasal administration. And then really the key thing for us was then also taking that into a larger animal model where we, of course, have much more overlap and similarity when it comes to the intranasal space. So in a non-human primate study with an H1N1 challenge, we saw exactly the same, which was really a wonderful way to kind of round out the story. So when we gave a huge excess of H1N1 virus intranasally, that daily administration with our lead asset really provided protection there. So having basically no chance for the virus to replicate in that upper respiratory tract and providing really, protection there at the gate from the get-go.
So, Koenraad, great results, a great paper. Does this validate the approach moving forward then? Are you ready to move into human trials?
Yes, going further into human trials, and yeah, this was a key. This was the most important step that we took with the company so far, where we really now showed and proved, that our the approach really can work, and both in animal data, but also very importantly in the clinical in the human data. Luckily, Dylan, you said that we're both on the paper. Luckily, I'm not on the paper. There were people that have much more expertise and much smarter than I am. Our scientists, of course, such as Clarissa, but also many others, and also our partners, where we worked together with to achieve all these results. Now, this is for us is very, very important data. Where Clarissa already mentioned it, right? But we see it's well tolerated in humans. We achieve these high antibody concentrations also in humans, and we can match that with the animal data that we can there protect these against all these different viruses. So really showing there that proof of principle, and that these antibodies also still intact once they have been in the noses, and we have many different molecules, of course, in our nose, in our throats, that actually break down molecules. We see that they are still intact. So that was really key data. And with that, we're continuing indeed, our later stage clinical trials now to further work, towards on the regulatory pathway there forward. And that's specifically, of course, for what we call PanFlu, right? Our flu program based on the CR914 antibody. But it also validates the broader approach of using these monoclonal antibodies administered through the nasal sprays. So yeah, very, very important for the company and very exciting results.
One of the other exciting things for the company is always securing funding, and last year you secured another 50 million in funding there. How is that going to support your pipeline moving forward? How is it going to support this platform?
Yeah, no, very important indeed. And we had multiple funding rounds. Last year, first led by a Singaporean fund from Temasek. But then also complemented with US funds and our existing investors of around 70 million. And then later on, we had also 30 million from the European Innovation Council, the EIC, and the Dutch Tech Fund, InvestNL. And then separately also 20 million euros from the European Investment Bank, the EIB. But it was actually funds that came from HERA, and HERA is the Pandemic Preparedness organization or initiative of the European Union to, also make sure that, we protect ourselves against the next pandemic. So, as Clarissa also already mentioned, we are developing these nasal sprays for seasonal viruses. We're now in the midst of a flu season, both, at least in the Northern Hemisphere, so both in the US, but also here in Europe, where I see also many people around me, unfortunately, having flu. And what we often forget that there's, people that are healthy that would have maybe, a week of disease, but people that have other kind of diseases, they really can end up in the hospital. You see now hospital wards that are fully focused on with flu infections around these times every time. But then separately, and where we got this funding through HERA, is also to protect ourselves against a new pandemic. So whether it's avian flu, bird flu, or any other flu, that we also have that ready to protect ourselves. And there we're also very, very happy with that funding through the EIB from HERA as well, to specifically focus also on that pandemic preparedness. And we're doing that here in Europe, but also across the world. For example, in Singapore, we also have discussions on how can we protect, in this case, the Singaporeans. That's a country that typically looks really, far ahead and trying to already guard itself against, future threats, and doing that in a very rational way. And there, we are also having discussions. How can we make sure that we help there to protect against the next pandemic, and that the economy can stay open while people are protected against a new virus?
So in a sense, can we say that we've learnt something then from COVID? We're going to be better prepared next time?
Yeah, well, I hope so. We of course, as a company, absolutely. So just thinking about it, when we suddenly got ourselves into the pandemic, I think nobody could have expected the way it turned out. And then the amazing response that came up also with trying to develop vaccines in a rapid time. But what we have is then nasal sprays that can protect against all these different viruses, where you can actually have that ready before there's a pandemic hits us. And then it's also a nasal spray, so that's easily self-administered. So you can then ship that to people's home once there's an outbreak, even before there's a pandemic, and then have people protect themselves against these different viruses. So then we can just keep the economy open while protecting people against such a new virus. So that, in that sense, absolutely would have a key new tool in the arsenal to protect ourselves. And I really hope that also across the world that governments also think about it and in that indeed not forget where we were in 6 years back.
Koenraad, looking forward in these next 12 months, what sort of milestones are we looking for now for PanFlu, but also the broader portfolio that you've got there?
Yeah, so with the data that Clarissa also just further explained, key data that we now got, now we're bringing, for PanFlu, we're bringing that forward into the larger scale, phase 2 trials. So that's we're now working hard on to have those started, but then also broadening. We have also broadened the approach where we also have an antibody that can protect against all coronaviruses that we see from the work so far, and then also broader than that. So progressing there from the exciting research we did, also other molecules into development further as well.
Clarissa, if I can ask you, where do you see this field of mucosal immunity evolving towards, and what sort of role is Leyden Labs going to play in preparing for that?
Yeah, I think, again, very much, I think maybe also echoing what Koenraad mentioned earlier, but for me, what we've seen, really in our own research goes beyond flu, right? I think we've really sort of understood now that taking this intranasal mode of administration for any virus that really uses that as a portal of entry really is really sort of an untapped niche. So for us, indeed, very much this is really the direction we're heading. So understanding, indeed, also as mentioned in the context of coronaviruses, like how can we also really start to build up that program? And I think important to note there is we also have this PanCOV asset that really goes beyond just SARS-CoV-2. I think we, again, are very much still, of course, aware of where we were 6 years ago, where COVID-19, of course, was the SARS-CoV-2 pandemic. But again, the coronavirus family is broader, right? So again, we have the same basis as we formed for our flu program, where we want to have a candidate that can really kind of be prepared for any type of coronaviruses or, any sort of mutation within that family. So we really want to see that through as well. And I think, again, we're really, looking in our platform to really sort of take this forward as a mode of administration for any type of respiratory viral infection. So that's really where I hope to see, us advancing over the next couple of years.
And Koenraad, what is exciting for you as we're heading forward into these next couple of years? The impact of these nasal sprays, the capacity to respond more quickly in the case of a pandemic with something that's already got broad protection. What's exciting for you when you're looking forward? Can you see a really bright future?
Yes, I can, and I'm an optimist in general. Now we have, of course, a lot of work ahead of us, but I'm very excited to do that together with a fantastic team. Clarissa leading all our research activities, but also with the other people in the team that we're doing that together. But what excites me most is really being able to protect people much, much better against these viruses. So every year there are 5 million people ending up in the hospital because of flu, and up to 650,000 people that are dying from flu every year. So that's really big numbers, and for a disease that should not be the case. Vaccines are very important, but for respiratory viruses, for airborne viruses, they're not enough. And we see them consistently, having a low efficacy, especially if you look at flu. So there's a really big unmet need to help protect people against just a yearly flu in a much better way, and to stop these viruses already when they're starting replicating in our upper respiratory tract. But there's no good thing about that. And then secondly, indeed, let's not hope. I hope for that there's not going to be next SARS-CoV-2, or like that, for example, bird flu is becoming more infectious to humans, so that it causes a massive problem. But if we're in that case, that we have something that can protect from very early on, so that we don't find ourselves again in 2020 and having to close down everything from schools to restaurants to offices, because we have to protect people against that. So protecting the people against these different viruses is really what excites me, and I'm really privileged to do that with a fantastic team here at Leyden Labs, both here in Leiden, the Netherlands, but also in the US and in Singapore there as well.
For people in the audience interested in learning a little bit more about Leyden Labs, where can they follow the progress? Where can they explore working for you? Where can they read this recent publication as well?
We try to stay current on our website, LeydenLabs.com. And of course, always feel also free to reach out, and we are also trying to be, of course, at conference and that kind of things. But at the website, LeydenLabs.com, I think is the easiest.
Koenraad, Clarissa, thanks for joining us today on Beyond Biotech.
Thank you, Dylan.
Thank you.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.